Hello, my project is titled Identifying and Characterizing Gene Co-Expression Modules Underlying Resistance to Androgen Deprivation Therapy in Prostate Cancer. The transition from unicellular life to multicellular life was a key evolutionary event requiring cells to coordinate and communicate with each other to ensure an organism's survival. This new cooperation was achieved by acquiring new genes forming gene regulatory networks to coordinate and regulate these processes. Previous research has shown that regulatory networks are often dysregulated in cancer, allowing tumors to survive and progress. This dysregulation subsequently results in many of the hallmarks of cancer, which are a key set of features common across all cancer, which become more pronounced as the disease progresses. However, another way we can look at cancer is as tumors losing features of multicellularity, expressing more unicellular features as the disease progresses. This is best explained using the atavism hypothesis of cancer, which describes cancer as the failure of fundamental cellular processes, reactivating primitive transcriptional programs that evolved in more ancient unicellular organisms. A notable example of multicellularity loss is found in prostate cancer, where primary tumors are graded using a histological Gleason grade to measure tumor dedifferentiation, which is correlated with a worse prognosis. Previous research within our group revealed as prostate tumors increase Gleason grades, they preferentially express more unicellular genes, likely due to a coordinated breakdown of gene regulatory networks required to sustain multicellularity. This coordinated breakdown may have prognostic value, enabling us to understand the underlying mechanisms driving prostate cancer. By building on this, we aim to characterize how gene regulatory networks change from normal prostate tissue to form tumors, and how these gene regulatory networks then change at multiple stages of prostate cancer. This will enable us to identify gene expression signatures of tumor behavior, which has immense clinical value. I will use a computational approach to develop an evolutionary network analysis pipeline to characterize changes in gene regulatory networks across multiple stages of prostate cancer. Using gene expression data from prostate cancer patient samples, the pipeline will first identify genes co-expressed across tumor samples. Next, it will characterize gene ages and then perform network topology analysis to understand how genes regulate each other, focusing on connections between unicellular and multicellular genes. And finally, it will perform functional enrichment analysis to understand how activated pathways relate to co-expressed genes. This study presents a new paradigm in prostate cancer biology, which aims to understand how gene regulatory networks maintaining multicellularity are broken down in prostate cancer, driving tumor progression by activating unicellular pathways. If you would like to know more, please have a look at my poster through Slack or send me a message. Otherwise, thank you for watching.